So I think I would prefer the section to be more practical than theoretical. I think that's when we give more value to we give more value to the presentation. So I don't need to introduce myself since Rachel has done that one. So we begin directly. The topic is just data visualization, but we are not just going to be data visualization alone, because I check those people that have registered, and then some of them are graduate students and postdocs. So rather than just to do ordinary data visualization, it's better we do kind of like apply plant breeding data visualization, which will be more relevant. So for the outline, we have different file format commonly used in data analysis, especially applied to bioinformatics. We have bioinformatics application, different data, and then how we can visualize them in any of the platform, either Linux or app. And then we also run diversity analysis, not only the theoretical, we have chance to do it in R, which is purely data visualization, a data that we transform for a matrix to a figure, and then we can see. So we run the DAPC, PCA admission network analysis. I hope the networks may take some time, but I guess we have some time to conclude it. We also do some genome-wide association study, which is part also of the data visualization. Genomic prediction, I don't think we have time, but I just put it there in case probably for next feature we can look into that one. I think there are some people waiting, so probably they can just admit that. Okay, so moving forward now, we talk of the different formats that we have in data. Since we are talking about data visualization, you should have an experience, you should have a view or know this type of file when you have it before you start looking at it either in R or in any of the platform you want to use. So we have the FASTQ file. Generally, the FASTQ file is come from next generation sequencing after you sequence your file. And then the way to identify a FASTQ file is just by at the initial or at the beginning of your sequence information, you have this sign which is @ So when you have this sign at the beginning of a file, you know that you are dealing with a FASTQ file. And then from there, you have a lot of information embedded in the file. It's not, you can open it generally in Excel, is somehow difficult. So you have to use some either Linux, or you can just read a FASTQ that you can load to view it. And then these are kind of like different information you can see in a FASTQ file, unique instrument name, and then the flow cell, all those information, how the material were sequenced, the barcode information, all those information are embedded in the FASTQ file, which will allow you later to do some demultiplexing to remove the barcode, and then some of those information that you are not looking. So you should be very careful on this one so that you're not missing a lot of information. Then the second file we talk of it is the FASTA file. So the FASTA file generally the sequencing information also. Generally it can be a short sequencing, or can be also a long, whereby you have all the chromosome if the reference information. Hello? Hello?
Hi.
Okay.
Yeah, just go ahead. We can hear you.
Okay, perfect. Then you have this is the way the FASTA file looks. So at the beginning of the file, you will have this greater sign just here. Then you know you are in presence of a FASTA file. And then after this one, you have the header line, and then the name of how the sample were named, and then you have the description. So the description, you have all those information, how the mapping, where was this one coming from. Then you have the sequencing information. So generally the FASTA file, you may see what we call reference file or genome information. And then the extension is finished by .fasta or .fes, .fa, .fna, or .fsa. So all this one, they were kind of like what we call a FASTA file. So please, if you have a question, you can just probably raise your hand and then someone can help me to monitor the discussion. Then we have the most common file that's in data visualization or data analysis or in bioinformatics. A lot of people use it, what we call variant call format, commonly known as VCF file. So the VCF file here now is you have your FASTQ file, you have done your mapping, you remove, you do some trimming, you remove all those barcode information, do your alignment with your reference genome, and then you pass through some intermediate phase, the BAM, and then the SAM file, you will end up with a VCF file. So the VCF file is somehow very comfortable, very easy to manipulate, and then and then the way to identify your VCF file is you have this hashtag 2 at the header, and then you have also some information. You have the chromosome, the position, the ID, the reference allele, and then the alternative. The reference allele information coming from the reference genome, while the alternative coming from the sequence the file you sequence at a physical at a certain physical position. So we have also some intermediate file with the BAM file, which is generally the binary file, and then we have the SAM file also. But generally, this file, they are just intermediate file. We don't use to store some file either as a BAM. Is in some very rare occasion, you can store some information in the BAM file, except if you want to load it in some specific visualization tool like tablet or flapjack where you require some BAM information. Now, let's move in now to kind of like a quick discussion on bioinformatics and then how we can use this one for data visualization. So generally, you have this phylogeny analysis. We do it today, yes. And then we have sequence analysis whereby you have your FASTQ file, you can do your alignment and see how your sequence are aligned at each physical position. You have SNP and the SSR identification and still useful in some lab, drug approaches, next generation analysis, and then metabolomic system biology. So bioinformatics is just a way to summarize this one, that bioinformatics is just a tool being used in different domains. It's applied in different environments today. So good. Now, data visualization at IITA, I focus generally on YAM because I'm working on YAM. So all the data, all what I'm going to present here is for YAM. So it's my research. So I have a right to present it. So we have genotype a lot of files whereby we just pick some sample today and then we run the analysis and then we see how it works. We have done some whole genome sequencing whereby it's kind of like very heavy data, 333 sample, and then we have some GBS data also. Diversity array technology, Intertech for different purposes. So those are kind of like different files that we load in R and then see how it looks. And then this is kind of like my routine activity at some time if I have some jobs and then how to perform all those activities. So this one used to help me, especially from diversity to genome-wide association study. This one used to help. So here, as I was saying before, you have your FASTA file here, and then you do your self-alignment, and then you can end up with pairwise and alignment to see some complete genome and how your genomes look like. Then you have your raw read. If you proceed with some next generation sequencing, and then you end up with your FASTQ file to do your mapping with your reference genome, which is the FASTA file. And then from there, you proceed with the SNPs coordinates, which will allow you to identify your SNPs. Once you have your SNP here now, you can proceed for different analysis. If you want, you can come for QTL analysis. You can do phylogeny analysis. You can do complementary CDS analysis, and then you can do some tests for selection and also some evolutionary analysis. So this summarize kind of like a visualization aspect where visualization aspect that can be done in your with your data.
Please, sir.
Now, yes, please.
Yes, can you go back, please? Please, what exactly CDS phylogeny trees study?
Okay.
Is concerning, yeah.
You
what
have
is the
your
difference?
Okay. You can extract your DNA from any part of your organism. So it can be just either for the leaves, any part. And then you have the CDS. The CDS generally stayed for complementary DNA, if I'm right. Yeah, I think I'm right. Which generally can be extracted from cytoplasmic organisms. So generally, it's kind of like you are not looking at the entire you are not looking for the DNA from the whole plant, but we are focusing on some key aspects just to retrace just to retrace some evolutionary aspects. So it's not based on the entire DNA, but focus on some complementary DNA.
Okay, thank you so much.
Now let's move to the data visualization in YAM. This is purely partially from, we use some Linux coding information, but the whole analysis was done in R. So the first one you have here is what we call generally the network analysis. So the network analysis here, it helps you to do a lot of things. You can use this one to identify which of your organism or your species are kind of like the oldest based on the routing information. So here we have a lot of species. We have the breeding line. We have Alata, Kinesis, Quincylis, farmer variety, and then we are just trying to see here. We are just trying to see here which one is kind of like the route from where most of the recent breeding material were generated. And then here, if you look here now, you can see that most of the breeding line, they are in center, whereby you have most of the, what we call wide related, they are at the extreme initial, which means they are the root for a lot of breeding line, which is what we are expected. You can also move to principal component analysis here, and then if you collect your material from different country, you can use this one just to test them also to see how they are related. Do you have some gene flow? And then this one also can give you some idea about if probably some material been kind of like restricted to some country, or you have some sharing gem plasma between between countries. So the visualization gives you a very quick observation of what you have in your data. Then you have also what we call discriminant analysis, DAPC. We run them also, we run this analysis today also in the area. Then you see how the code works also. So here now we perform this and then we look at factor 1 and factor 2 in discriminant analysis, and then you can see that we have a very clear, we have 4 groups that are well distant. And then with this one now, you can perform what we call a molecular variance analysis just to see how the diversity vary within and then among the different clusters you have. So you can perform a lot of tests, either fixation index. You have different test analysis that can be performed for this one to confirm the level of the diversity. So please just stop me if you need some clarification that we can discuss. Then this is also another visualization in Flapjack. We can do this one in Python, whereby you just you have this one and principal component analysis. But rather than to focus on the S and Y, you can also factor the 3 axes and then see how it looks. You can see this one generally have your S and then Y, and then you can see how your sample looks in the space and then in the time also. The code is at the code for this one. If you are interested in Python to implement this one, also you can check on my GitHub page. The code is there for you can take the code and run it very easily. It's just a matrix data that you need, which if you have a factor, and then you just need probably to transform to scale the data, and then you can run it easily in Python. Then we have this one also, which is also a data visualization, which really help a breeder. This one is mainly used for breeder, either animal breeder or plant breeder, to make some selection, whereby if you want to develop a new variety or you want to have a new breed space, you can make a selection here. If you select some individual that are 100% pure red here, crossing with some 100 pure green here, so you know what you are expecting. But if you select some individual here with half red, half blue, and half gray, it's too difficult for you here now to start looking at what happened during the progeny, during the offspring. So this one gives you a very good, give you a picture of what you have, and then how to make the selection of what to cross for the next generation. So another visualization that we'll talk today is genome-wide association, which is kind of like just to look at the genome and see which part of the genome or the chromosomic region are responsible for some key traits. So we tried that one also. We have different models that can be used. We have the KQ model. We have the ordinary Q model, whereby you just factor in the population structure as a factor. You can also make use of the kinship matrix. So the kinship matrix and then the population structure can help you to correct your population to avoid some false positive discovery. It's really important to look at those ones. But the good thing, probably before you start jumping into different models, you can just try to run the ordinary model without any population controlling and see how it looks, and then use the QQ plot to confirm if it's overfitted or underfitted. This is the kind of the simple model we have here, whereby you have this one, which is the ground mean stand for the phenotypic. You have the marker effect, and then you can control whatever you want here. Yeah, so this one also is visualization, whereby if you have your markers, you can just check the distribution across your chromosome. And this is the case of YAM, whereby we have a 20 YAM chromosome, and then we look at the density. This one helps you just to know exactly what happened during your filtering. If probably some of those markers were, if you need probably to input your data, or some of your region are missing in terms of marker coverage. Because for GWAS, we are looking for good markers. At the same time, also, we are looking for good coverage. Because if you filter probably some markers from some region, and then mistakenly those regions that are controlling the traits, that means you are missing out those information. So you really need to be careful when you want to do some either GWAS. But for diversity, we don't care much. But for GWAS, you need to be really careful so that you not miss out some good marker information. So this is the GWAS using different model, whereby we have the dominance, and then we have also the additive model, whereby we tested different approaches. And this is some of the paper that we have published in the YAM, so you can read more about it if you are interested. We have chance to run also the code today, and then you can see how we fit all the model. So this is the continuation. Now let's have a kind of like a quick discussion on the genomic selection before we go to the practical aspect. So the aspect of the genomic selection is kind of like deploy the phenotypic data combined with molecular information to see how best we can predict the performance of some progeny. So we have what we call the training population, and then we have the testing population. But in the case of yam, the way we are applying our genomic selection is for parental recycling. So we predict the performance of the progeny by the combination of two parents. So if we cross A and B, what are we expecting? In that case, it gives you an advantage to select those parents that have high crossing ability. So, you know, if I cross this and this one, I'm expecting a progeny with high breeding value. So that is how we proceed. And then this is kind of like a testing that we have done in the yam, whereby we combine rapid seed multiplication, combined with markers information to run our model. This is the process. You have this one, and then you make this one. You go to the field, you select your cotton, go to the SH, then you plant them in field, and then this is one of the field. And then the main importance is here is how we fit our model. But for a crop like yam, when you want to do a genomic selection, we don't only focus on additive alone, because you want to visualize everything, want to see the performance of every single thing in the potentiality. So you not only focus on the additive, but you focus as well on you consider the dominance in your model. So here we use the ASRM, and then we focus, this is for yield, and then you have your replication, your poem, and then you have your accession name additive plus the dominant. So this is basically what we did, and then we select our parents, and then this is the selection and and then the advancement, and then we're able to make some very good selection, move them to field, and then evaluate them in the field to see the performance of the progeny. I think, as I was saying before, I want this one to be kind of like more practical than we should just talk. So that's why I make the presentation very short. Then we now move to R. But before we go to R, please, if you want me to explain something, please feel free before I move to the R.
Thank you for your presentation, Dr.
Okay.
Paterne. Yeah, for me, it's not a question, but is to know if you can share after the presentation the GitHub link where you put all of the screen. Even I don't know if you have also screen on Python to do some analysis. If you can share with them, I will also be happy.
Yeah, that
Thank
one
you.
will not be a problem. Most of my code, most of my scripts, they are open, and then yeah, some of them are in my GitHub. I think I did not protect them. Most of them, they are open. The Python, I have not yet put them on the GitHub. I still need some time to kind of like clean them, and then I don't have much time for the Python to put them on the GitHub, but most of my R and then Linux code, they are already on the GitHub. Anybody can pick it for such analysis. So there's no problem. Yeah, I will share the presentation as well. There's nothing secret about it.
Thank you so much.
You are welcome. So please, should we go to the R or myself? I want to learn from all of you. So please feel free if something you think, okay, with this one can be done other way, please feel free. Myself, I'm here to learn from so I'm open for suggestion and everything. Yeah, so please feel free.
Let's go ahead and leave the question till the end so that we can maximize the time, since we have 1 hour and it's already like 30 minutes gone. That way we can test you. So maybe you leave the last 10 minutes and we can entertain questions from, the people who have questions.
Okay.
Thank you. Yes.
Perfect.
Thank you so much.
So the first activity we want to do here is just to try the diversity analysis in visualization. So I will give you a background of what we have, because I don't want to start from VCF, go back to my terminal, open it, and then how we do the alignment. So here I have generated the blank information, what I converted to a matrix data. So the matrix data is what I'm going to load here. And then what we need here, we need this library, EP, and then is also available. Sometimes you may need some bioconductor because you have some code that are available there. We have this package that are very good also. This ggplot is very good for data visualization. And then the data is here that we want to use. I can also share the data also at them. And then I have generated my IBS data identity by state matrix, whereby I have factor in different factors based on some genetics information. So I will load the data. So I think, yes, so we have a 194 observation and then 126 marker. So here the 126 we have here just the marker, which is the dosage 012. So we can just, I'm coming, let me just run the view for you. So it's just a dosage, so views. So let me use the head will be much better, I think. So that I will not load everything. Yeah, I think it will be much better. Oh, sorry. Yes, I want to see how I will just share the 1.6. So it's just 012 that we load here, then you can see how it's okay now. So it's a matrix 012 that I just scale here as I was telling you before, just to have a very good information. Then what I need to do here now, I need to change my row name because I did not declare what I have as a row name when I was loading the data. But if you want, you can add your row name here. So it depends which one you are comfortable with. So I'm declaring my first column here as a row name. That is what I do here. Then in the data, I have what we call the ID, and then I have what I call also the class. So the ID stands for the clone name, and then the class stands for the factor. It can be a country, different factors. Then you run this. Then I declare my variable as kind of like the rest of the information. Then you can check what do you have here now. So what we have, we can see what we have inside. So we have cluster 1, cluster 2, cluster 3, and then these are the different components. So our plotting or our visualization now will be based on this information now. So what I'm doing here now, I generated some file here. I just want now to combine them by just using this function, the cbind, to combine both the row name and then the one I declare as a variable. So moving forward here now, we scale our data, then we transpose the data, and then we generated the distance information. So the distance here now, we have different type of distance. You can use the NACE, you can use the IBS, you can use the Jaka distance, different type of distance. But here, just for the sake of the time to be fast, I just put this distance, and then we run this one. Then we will have this UPGMA. We have either UPGMA or neighbor joining. So if you are familiar with anyone, you can quickly use it. But for me, since we are dealing with molecular information, I prefer to use the UPGMA because we assume that all the sample appear at the same time. Then probably the end time is the same thing. So let's give some time. It's still running. I hope it should not take much time. Okay, so now we generate the UPGMA. So what we need to do here now, or we need to define our color. We have, if you look at the factor, we have 4 different clusters. So we assign the red for cluster 1, blue for cluster 2, green for this one, and then a black for the fourth cluster. Okay, so we define our color here. And then moving forward here, we don't need this one. So this one generally just to play with the font size. You can increase, you can decrease it. Then we run this one. The on route generally used to take time, but we can try it. Let's try first the radial or the font. Anyone? Let's try the font. Okay, scale. Okay. And then we can run this one and see how it looks. Yeah, generally then you can print it. If you want to save it, you can run this one, either JPEG or PDF, you can save it. I will share this code with you also. So you can. Let's see, we have plenty information. So we can zoom it when it's come. Oh, sorry, yeah, the sample is quite much. Yeah, so this is our first visualization. So when you see this one, you can see that it's too kind of like the clustering is too much. So the fan or the radial will not be kind of like the best option for you. So you can now say, okay, that is where the visualization coming in. Now, what can I do to change this one? Then you can go back and say, okay, if I run the unrooted may be much better for this kind of analysis. So you run your unrooted, but this one used to take time. Yeah, the unrooted used to take time. What happened for the unrooted, it has to decide which one to be fixed and then how it's going to fix it. So it take them each sample and then you run what we call the boost wrapping. So you consider them running several times before you could decide which one stand as kind of like the appropriate route for you. This one may take like five minutes before it's finished to run. Generally, such analysis, if it's heavy, you can put it on your server. If you have access to server, it runs very fast. And then, yes, on this, because you can partition your server into multiple core. But yeah, so that is generally the best option. So let's wait for this one. So when this one finished, we can move to the second aspect. So yeah, sorry, this one takes some time before it finishes. While we are running this one, I can be answering some questions. So we are still waiting for this one to run.
Yes.
So please, feel free.
Yes, thank you for this presentation. My question is about the group. You show that you have 4 groups now, and you show that you have around 400 or something like this per group. So how to have the list of this group, the accession, if it is accession or the samples per group, how to import this information to do
Okay.
after your.
Okay. When we run the discriminant analysis, you can see how we can get those list from. So we have this one. Before I could factor into the matrix, I first did the DAPC and then the admission. So those one give me kind of like each accession in the cluster they belong. And then from that one, I use it now as a factor to plot my hierarchical cluster here. So when we run the DAPC, I will show you how you can get those information from that aspect.
Okay, thank you.
Okay.
Okay, I want to confirm, is it that for every visualization you do, you have to have it in matrix form before you could have any visualization done?
No. It doesn't necessarily mean need to be in a matrix. It can be a data frame, but the only thing that you need to be to be very sure. The data has to be numeric. The data has to be numeric. So you need to avoid either probably integer, character, all those ones. You need to avoid them. If not, the code will not even take it. But matrix is fine, but it's not compulsory.
Okay, so if the data are all numeric, is it that there won't be need for you to specify which is the factors from those which are not the factors, which are variables?
No. You don't need in this case. But the problem here now, you see, if we move back up and then in the code here now, you will see that we declare, yes, we declare this one here. So you see that we declare this the class as the class, and then we declare the ID as a call as a row name, and then we declare the class as a factor to avoid the confusion to allow the code to run properly. So we declare this one, and then later the rest we declare them now as a variable, and then we scale them. Because you can scale the class something like cluster1, you cannot scale it. It is not a number. We only have access to scale a number. I don't know if you get my point.
Maybe I understand, but I just need clarity on point again. What I'm saying is that whether we make it in factors, like define a particular variable as factor, or leaving every of variables as numeric, it will still run.
Yes, it will still
run. Or we
run.
just make it numeric.
Yeah, it will still run if you have them as a numeric.
Okay, then finally, please, sorry.
Yes.
Finally, is it that with these three package, which is the Phanogram, GG3, and ggplot2, we could do a whole lot of visualization?
Yes, you could do a lot of visualization. There are a lot of packages that it depends of your area of expertise. If you are probably into a data scale or economics, you have your own package that you can use. But if you are into a plant breeding, probably genomics, you have a G45, you have this ggplot, you have a Fungum. They are kind of like almost like 10 to 15 packages that you need to be really familiarized with. I will put the list of those packages that kind of like require for a very good data visualization. Just take your matrix and then transform it into something that can really be visualized. Yeah. I will put the list of those packages also, so you can check the metadata and then the vignet also.
Thank you very much, sir.
Sorry, it's still running, so I think it's good. So we can be answering the question when this one finish. We can move fast. The rest is not.
Okay, with regards to font size, is there a specifics? Is there really a specifics that you would get to, or you just get to keep trying changing numbers to see one that will give you a very good outlook?
It depends generally of the number of samples you are dealing with. If probably you have a few samples, you can increase it to the maximum, which is 1:1. But imagine you have a 2,000 sample and then you put it 1:1, you will not even see anything. So you play with, it depends of the number of, and then also during the visualization when you are running, okay, this one seems good, I need to adjust this one. So it's come also with your taste and also how you, yes. And then in the code here also you have where you can play with either bolds. Yes, you have a way where you can play with bold, adjust, all this one, the size. So you can read the vignet of this code to see exactly how this one is being scaled. Yeah, I just want to show you the on routing, and then you will see how this one improve just have a very good look at this, and then you will see when we run the unrooted, you will see how it's improved the quality. So when you have different for when you are writing a paper for data analysis, you need to be careful of then which figure to use to represent exactly what you want to achieve.
Hello.
Yes, please.
Yes, sorry. Here it's evening. So
Okay.
my name is Eris, and I'm sorry I joined in a bit late, but maybe as we wait for the run,
Yeah.
just requesting you to recap on what you're clustering, what data are you using, and what are you clustering?
Okay. So here is the data generated from genotyping by sequencing is 2,000 sample. So we have the FASTQ file, and then we proceed with alignment, all those one. We got our VCF file, and then we run a lot of analysis using my terminal here. So let me see if I can see if it's possible to see my, yeah, let's see. Yeah, so this is generally some kind of analysis, but we don't have much time. So from the VCF file, you run what they call the dosage file. So the dosage, you take your data from the VCF file, and then you run the dosage. So the dosage is just, I think, VCF file, dosage file, and then it give you the 0, 1, 2. So the 0, 1, 2 depend also the level of diploid. Since this one they are diploid, so we have the 0, which is for homozygous reference, 1 for the heterozygosity, and 2 for the homozygous alternative. So the data we are clustering here is what we call dosage file. So the dosage file is 0, 1, 2. Then we now load it into the R using different packages. And then we generate the distance. So from the distance, now we run the clustering based on the shared allele common, what they are sharing in common. Both this one is taking my time. Sorry, yeah, I generally run this one on the server because we have our R on the server, but I'm not seeing in Nigeria, so having access to our server is somehow complex, except I turn on my VPN and yeah.
So generally, these clusters are showing the homozygotes, the heterozygotes, and
No. We are not, this one, you are not seeing anything concerning the homozygotes or heterozygotes here. What we are seeing here now is how the genotype were related. Is the relationship matrix that we plot to visualize. Because if I open the matrix, it will be too difficult for you to know what is this matrix say. But when I load this one, you can see that the red, they are kind of like well classified in one cluster. But when you take the green, you can see that the green, they share a lot of allele common with the blue and then the black. So we don't have any information about the heterozygosity, but here now we are just visualizing, the relationship among the different genotypes. So you can say, okay, we sequence this one, the genetics matrix vary from so to this one. The high genetic distance was observed between this sample and this sample. This is what we are just trying to represent, because it's just a visualization information.
okay, so it's basically showing genotypes that are related, that are close related, and those that are far from.
Exactly. You got the idea. Okay,
Okay.
I think probably we can let's just continue. So we have this one, then we can, I think we still, it's still running. I really want to, let's give kind of like 1 more minute to see if it can come.
Okay, meanwhile, I can ask something again.
Yes,
still
please.
Concerning the relationship between the different genotypes. Usually I know that, okay, not that I know, but I hear that when you're making such visualizations, you specify the root distances. I don't know how to call them. How to specify the distances that you would want to see. And how I wanted to find out, how do you do that? Or how did you do that in using your code?
Okay. In that case, if you want to specify, that means you have already some pre-information, but you collect your information from probably nothing, and then you don't know anything. So you use the UPGMA, which assume that all the samples were collected at the same time, and then they have equal chance to appear. So that is the arbitrary information you set in the code. Because if you, let's say if you have information that, or you have a checks in your material that this is the oldest, you can put that one and you fit it as kind like a control. But in this case, we don't have any information. So you use the UPGMA. Then the UPGMA treat all the sample as the same information. But the neighbor joining, if you use the neighbor joining now, you have already the scale. And yeah, you have the scale which will tell you, okay, this sample is likely to be the first one. This one is the kind of like the oldest one. So generally you can proceed with that one. But in our code here, you have this one whereby you run your UPGMA directly here. And then also you scale your data here. So by scaling, you give chance to all the samples, all the genotype to have the same the weight. Like the way you have 0, 1, 2, you give them chance for all of them to become like, after scaling all of them, they become like 0 point something. So based on that, now you can be sure, okay, yeah, it has finished finally. So based on that, now you'll be sure that you are not probably giving chance to any other thing. So you can see here now that this one is more informative than the first one we view. So you can see here now. So that is why I was really insisting on to visualize this one. So for this one, you can see that it's much better than the first one. Yeah. Please, someone is raising his hand. Yeah, please feel free.
Yes, thank you, dear communicator. I'm very excited to be in these sessions and I'm learning a lot. My question is about the different methods. I hear you use UPGMA for the clustering. And yes, my concern is it possible to run Bootstrap and or Bayesian model to perform the clustering?
Yeah, you can use the Bayesian model, but if you want to use the Bayesian model, yeah, yes, you can use the Bayesian model also. But the Bayesian model, that means you have to kind of like predefine. Yes, you have to predefine how you want to view it. The bootstrapping is also possible, but the bootstrapping, if like the 10K fold, you will run it for a very long time. And then for a training like this for 1 hour, will not be, I think the bootstrapping will take me like 25 up to 30 minutes before it finish. That's why I'm just, but you can use all of them. I think in the code also I have a bootstrapping, and yes, I have the bootstrapping also in the code, but okay, I will not be able to run that one. So it's possible.
Okay, thank you very much.
Then, you are welcome. Then the next visualization we do here now is kind of our data, and then we have the chromosome. Let me show you how it look, the Excel file, and then what we are going to plot now.
Okay.
Some idea, I don't know if it's this one. Let's see. Yeah, so you see this one now this is what we are going now to visualize in R. So for you have this one, so it's too difficult. You have now to start looking at probably all this information. But very easily in R, you can just do like this. So what we want to do here now is just to plot this one, we load this. Then we can see, so we have already the distribution on the chromosome. So you see how R is very useful. So you have your markers, then you can easily look at how the distribution of the markers. Then, so you can plot this one. So we can check with our plots this one, we can see, I hope it will be there. Let's see. Okay, this is our marker density. So you can see this one. So you see here we have kind of like a lot of information here. the high density here. So this one gives you some information that you need to focus on this chromosome 9 to see what's happening at this one. There are a lot of kind of mutation going on here, probably some region that are still under evolution. So all those things, when you have this one, it gives you some quick information how to do with it. So yes, now we can do the DAPC, and then someone asked how to assign the cluster. Now we can now run this one. Generally, the DAPC doesn't take time. So we have this is the raw data, the dosage format, which is 0, 1, 2. And then the next thing, we convert them to a data frame. Then the package we use this one to generate the raw file is for human being. And then for the human, which is plenty, we have the sex, we have the phenotypic. But here it doesn't concern us. So we remove those 1 to 1, 6. That is what this line of code they perform. So you can write the dosage if you want to use it for something else. Then we define it as a matrix. And then very important information for the DAPC, DAPC doesn't accept missing information. So you always check this one 0, which means I'm okay. Let's see, I think I should upload this one. Then we run this one. So we just want now to define some markers information like from 0 to, it's kind of like a PCA, but we are forcing the information not to be arbitrary. We can plot this one easily and see how the marker looks like. So we have 0 and 1. So this is how the marker looks like. Then next thing now, we just generate this one as kind of like a structure information. Let's see. So we run this one. Yeah, so please just be careful here now because it's becoming more complex here now, because here now we want to start assigning the group. So please, I don't know if Arno is still there. When you ask how we assign some individual to a group, so we perform some here, and then you will see.
Hello?
Yes, I can hear you.
Yes, I'm there. So
Okay.
can you repeat, or you are asking me a question?
No, I was not asking. I was just telling you just to pay some attention here.
Okay, great. Thank you.
So here now we start assign some individual to each group here now. That is what we want to do. So the BIC will give us some kind of like information on how many group or tendency. So you see here now we have 1, 2, 3, 4. So here now we can start saying like by 4 we have a kind of like a click. So this is kind of like we have a subgroup here, but the main groups happen here, which is around 10 group. Because this is where you have your final information, the good sharp elbow. Yeah, that is how they call it. But here now we can just say, okay, let's test for 4. So we test for group 4. So the BIC, which stands for Bayesian Information Criteria, help you to kind of like have a very good information. So we have, this is how the sample were divided. Then we don't need to view it, so we can do our summary. And then we have this thing, what we call this. So since we are using 4, we change this one to 4. So you need to be very careful here. So you see now you can now write this one out if you want to see the individual. So by writing this one out, you can go to your file, to your directory, and then you see each sample in which group they belong. That was what you were asking for before. So you can see each sample group they belong.
Yeah, okay, exactly. Yeah.
Okay.
Okay. Perfect. Thank you.
You are welcome. Okay. So what we want to do here now, you now see, we explain the
So sorry. So you have also discrete on your GitHub, or you need to
except I check. I don't know if I have dropped it, but
this one.
general most of my code they are
Okay.
general, but I will check just to confirm.
But so especially it will be, I will be happy to have this one because this function I don't have it in my screen.
Okay.
No.
Hi, Dr.
Yeah.
Thank you so much.
Yeah, please.
Yeah, we're actually running out of time. I don't know how much you have left to present to
Yes, I think
us.
I'm almost done. Let me just at least finish the DAPC, then we can close.
All right, and then we have a few questions.
Okay.
Amaka probably would ask if there's still time, or we might ask, we might have to email the questions to you, so
Okay.
you respond to them via email, please.
Okay.
Yeah, thank you very much.
No problem. So I think I'm almost done. So here, this is very important to have to check. You see now the assigned probability. The assigned probability now for group 4, we assign them 100%. Group 3, we assign them at 99%. Group 2, 100%. Group 1, 100%, which means we don't have any kind like doubts about the individual being assigned to each group. So it helps you to have a, so I can just move to the final cluster. So my color, let's see how it will look like. So I can just plot this one then. So yeah, so this is how it's look like. So you can change it for different, yeah, sorry, let me take some question. The code seems to be much, but 1 hour will not be, to be honest, 1 hour will not be enough, but you can just have a kind of like a taste of what's look like on data visualization in now. So I think that is briefly what I can share. So probably I can take some questions before the rest should be mailed to my yeah.
Okay, I think they told you guys are going to ask.
Yes, you're already going to back.
Yeah, we are back from the forest and we want to go back to the hotel.
Oh. Okay. Yeah, so even myself, I think I have to leave now. My colleagues, they are ready to go. So but really sorry. Just try to email me the question, and then I will answer as fast as possible.
Okay, so are you able to take any of the questions now, or you really have to go at this point?
Okay, probably I can take some few questions.
Okay, I think we had somebody in the chat that asked, how about number of markers and genotypes for accuracy of dictation?
Okay.
I think the person was trying to understand about that, yeah.
Yes, I have a work also on that one. So if for what we call a quality control or quality assurance, you need to have something like a 50 to 100 markers if you want to do quality control. So 50 to 100, you are okay. But for genomic selection, the number of markers we retain is around 2,000, 2 plus, like 2K SNPs markers. So the number of markers is really important, but you can, for dictation, you can have some what we call the curve. So with the curve, I think we have it also here. Let me see with this. Yes, like for this one now, you have the number of markers. So for almost 1,000 SNP markers, I can explain 100% of the information. So I don't need to use all the markers. So all this information guide you to make the decision.
Okay, maybe you can stop sharing your screen and we can just take a quick picture.